Hi there. This video introduces how to import your data files from TSE Systems PhenoMaster into CalR for analysis. First, under the Input tab, select the drop-down menu under Pick the Template option to determine how many groups are going to be analyzed. The example file provided is a 2-group experiment. So for this drop-down, let's select 2 groups. Look at the box on the left labeled Import your indirect calorimeter data. Under the label 1/Browse Data CSV Files, you will see a Browse button that you can use to select the CSV file that we are going to analyze. Once the uploading is complete, you can download a CalR file by pressing the CalR File button just below where we imported the indirect calorimetry file. The standardized CalR file enhances consistency, speed, and reproducibility for the analysis of metabolic experiments. The next step is to describe details about your experiment. Go to the right side of the screen. This side includes all the metadata that describes photoperiod, body mass, and body composition. Fill in the times when the light cycle starts and the dark cycle starts. In this experiment, the light cycle starts at 6 and the dark cycle starts at 18. Then, you can choose whether to import weight data. If not, weights from the calorimeter will be used. As body composition information will help with the interpretation of experimental data, the weight file also allows you to import lean mass and fat mass in addition to body mass. In this example, we don't have data of lean and fat mass, so select No where it asks you if you will include lean or fat mass. Go ahead and upload the weight file now. Next, you need to describe the groups. Enter the name of your control group first, and then enter the names of the rest of the groups. This experiment is a wild type versus knockout experiment, where both groups are placed on a high-fat diet. So for group 1, we will enter wild type as the first group name, and knockout as the second group name. Now, let's choose corresponding diets for the groups. For the first group, we can navigate the drop-down menu to select Research Diet 60 kcal%, fat 5.21 kcal per gram. We can do the same for the second group. However, for demonstration purposes, let's manually input the high-fat diet for the second group. We can manually enter this diet by selecting Other from the drop-down menu. This will create blank inputs that we can populate for diet name and caloric content. For the name, let's input Research Diet 60%, and for the caloric content, let's input 5.21. Finally, we can drag subjects to their corresponding groups. In this example, Group 1 contains subjects 940, 942, 943, 944, 945, 947, and 951. And Group 2 contains the rest of the subjects, which are 941, 946, 950, 952, 954, and 978. You are all set with your inputs, and you can click on the Time Plot tabs and get ready for generating plots. Once in the Time Plot tab, the first thing you should do is click on the Session File button to generate a CalR session file. This will create a file that captures all the work we just did and which you can use to upload to autofill out all the same experimental variables that we just filled out for later calorimeter file analysis. Next, you can click on the Generate Plot to generate a time plot and begin analysis of your indirect calorimetry data for your TSE files. Remember that if you are using your own TSE files, your experimental variables and input tab variables may differ from that of which we showed in this guide. I hope this was helpful, and as always, if you need any help, please make sure you reach out to the Banks Lab via the email provided on screen. Thank you.